Good morning, my dear students. In the previous topics, we have discussed about what is reproduction, what are the two types of reproduction, that is asexual and sexual, and the various ways in which the asexually reproducing organisms can reproduce. Today, in this video, I'll be discussing some questions from the NCERT textbook which have been repeatedly coming over years and are very important from the CBSE exams point of view. These are not so direct questions. Just think over them. Do organisms create exact copies of themselves? What is DNA? What is the basic event of all reproductive processes? How are variations caused? How are the different body designs created? Now we'll discuss the answers to these questions one by one. First, do organisms create exact copy of themselves? The answer is no. See students, in the previous videos I told you that asexually reproducing organisms produce exact copies of themselves. But it is not always the case, because even in asexually reproducing organisms, they produce a new copy of DNA before they reproduce. And during the production of the new DNA, there can be some errors which may lead to the change in the organism. So here we will answer as no. Biochemical processes are not exact, and their errors thus caused which alter the DNA and thus the body design changes. So there are errors which are caused during the DNA copying or other biochemical processes which can change the body design even in a asexually reproducing organism. Next, what is DNA? As you all know, DNA is deoxyribose nucleic acid. It is the information source for making proteins. So dear students, as you all know, DNA is the genetic material which is passed on from the parents to the offsprings. And it is the information source for making proteins. So new proteins which are synthesized in all organisms are made from the information which is acquired from the parents through the DNA. DNA is present in the nucleus of cells in eukaryotes, while in the prokaryotes, DNA lies directly in the cytoplasm. Now, what is the basic event which is common to all reproductive processes? The basic event which is common to all reproductive processes, whether it is asexual reproduction or sexual reproduction, is DNA copying. DNA copying means the parent strand of DNA undergoes replication. Replication is the process by which DNA produces two daughter strands, which are the replicas produced from the parent strand, in which one strand is the parental strand, while the other DNA strand is the newly synthesized strand. In this way, DNA forms two copies, one of which can stay in the parent cell and the other one can go into the daughter cell. Or in some cases, both of them can go into two new daughter cells. Next question. How are variations caused? Now we have discussed this point just now that in asexually reproducing organisms, variations are caused by errors during DNA copying because they do not have any other process in which there can be variations which can be produced in DNA. So it is only due to errors which may occur during DNA copying that can lead to change in the variations or changes in the asexually reproducing organisms. While in the sexually reproducing organisms, variations are caused due to three reasons. First, due to errors in DNA copying. DNA copying में जो error है, वो दोनों में आ सकता है. In the case of a sexually reproducing organism also, and also in the sexually reproducing organism. There is another process called as crossing over. Now, this crossing over occurs during the gamete formation in the sexually reproducing organisms, and this leads to changes in the DNA. Now, because sexual reproduction involves two parents, so by combining of DNA from two parents, variations can be introduced. Next question was how variations lead to altered body designs, or how are the changed body designs produced? Now, variations in the DNA lead to changes in DNA. So, variations की वजह से changes आ रहे हैं. और आपको अभी बताया था कि DNA is the information source for synthesis of protein. So, DNA अगर change हो रहा है, तो proteins भी automatically change हो जाएंगे. और proteins हमारी बॉडी के बिल्डिंग ब्लॉक्स हैं. इनसे हमारी बॉडी के स्ट्रक्चरल कंपोनेंट्स बने हैं. सो स्ट्रक्चर्स चेंज हो जाएंगे, प्लस प्रोटीन्स हायर ऑर्गेनिजम्स के अंदर हॉर्मोन्स का काम करते हैं, एंटीबॉडीज का काम करते हैं, एंजाइम्स का काम करते हैं, और अलग अलग तरीके से if there are changes in the proteins, then with the variation either in hormone or in enzyme or in the immunity due to antibodies or as a structural component, the body design will change. The functioning can change. Next question is, during division of cells, which process succeeds DNA copying? So हमने पढ़ा कि सबसे पहला प्रोसेस जो होगा वो होगा डीएनए कॉपिंग. डीएनए अपनी कॉपीज बनाएगा. तो इसके बाद क्या होगा? इसके बाद डीएनए बनने की जब कॉपीज न्यू सिंथेसाइज हो जाएंगी, देन देयर विल बी डुप्लीकेशन ऑफ द सेलुलर अपैरेटस. सेलुलर अपैरेटस मतलब सेल ऑर्गेनेलीज भी अपने आप को डेवलप करेंगी, क्योंकि अगर सेल ऑर्गेनेलीज न्यू नहीं होंगी, सो डीएनए एक organized cell को it cannot give rise. So the answer will be, during DNA copying, new strands of DNA are synthesized from the parent DNA. But this process cannot give rise to organized cellular structure. तो सिर्फ DNA से पूरा सेल नहीं बन सकता. Hence, formation of cellular apparatus by duplication of cell organelles and cytoplasm complete the process. So इसके बाद जो नेक्स्ट स्टेप रहेगा वो रहेगा हमारा डुप्लीकेशन ऑफ द सेल ऑर्गेनल्स. सो यही चीज हमने एक छोटी सी टर्म के अंदर पढ़ी थी, जिसे हमने कहा था कैरियोकाइनेसिस एंड साइटोकाइनेसिस. तो अब इस चीज के साथ आप उसे कोरिलेट कर सकते हैं कि पहले कैरियोकाइनेसिस मतलब न्यूक्लियस, और न्यूक्लियस में क्या चीज डुप्लीकेट हो गई? डीएनए. और उसके बाद क्या चीज डुप्लीकेट हो रही है? सेल. सेल डिवाइड कर रहा है. और सेल कब डिवाइड करेगा? जब इट विल हैव द सेल ऑर्गेनल्स व्हिच कैन कैरी आउट द सेलुलर फंक्शंस. Now, why is variation beneficial to the species but not necessarily for the individual? This is an NCERT question which is very important. Now, variations may cause changes in DNA. Variations की वजह से DNA में changes आते हैं which may not be useful. जरूरी नहीं है कि वो इंडिविजुअल को, जिसमें वो चेंजेस हुए हैं, जरूरी नहीं है कि उसी इंडिविजुअल के लिए वो बहुत फायदेमंद हो, लेकिन आने वाले टाइम में they can help in the survival of the species. For this, there is an example given in your textbook regarding bacteria. I discussed this example earlier also. So suppose there is group of bacteria living in a pond, and some of the bacteria or one of the bacteria has a variation because of which it can survive in the warm climatic conditions. So when the environmental conditions changed due to global warming and there was warmer conditions, all other bacteria which did not have this variation could not survive, but this bacteria which had this variation to survive in warm conditions could survive. So the individual which had this variation, जिसमें ये variation पहले आई थी, उसको उस चीज का फायदा नहीं हुआ. किसको हुआ? उसकी स्पीशीज को, क्योंकि उसकी स्पीशीज सरवाइव कर गई एडवर्स कंडीशंस में. Now, बहुत सारे students को there was doubt between, that is, what is the difference between regeneration and fragmentation. So just look here. In fragmentation, the fragments which are produced from the organism can grow into an individual organism. But in regeneration, generally, the organism grows a lost limb or other part of the body. A new individual emerges from each fragment. But generally, in case of regeneration, no new organisms are formed. In fragmentation, only a few organisms can fragment and form new individuals. Regeneration, all organisms exhibit the ability to regenerate to some degree. Even we, हम लोग को भी अगर चोट लगती है और हमारी स्किन हम रीजनरेट कर सकते हैं, हमारे कुछ हद तक लिवर को भी आप लोगों ने सुना होगा, लिवर भी रीजनरेट किया जा सकता है. So even we have this ability to regenerate up to some degree, but new organism is not being produced. Fragmentation is observed in organisms such as Spirogyra, as we have discussed, and in sponges, in animals, that is phylum Porifera. While in regeneration, we include the examples like growing of a lost limb by an organism like we talked about octopus, or the regrowing of the tail of a lizard. These are the examples of regeneration. So regeneration, we can say, is an accidental mode of reproduction. It is not a normal mode of reproduction. Planaria, which can grow into new planaria from the fragments from its parts, is also not showing this as a normal mode of reproduction. It can reproduce by regeneration, or it can produce new organisms by regeneration if it is accidentally cut into parts. Similar is the case of Hydra. Hydra reproduces by budding, but if accidentally its parts are cut, they can regenerate to give rise to new Hydra. I hope these topics are clear to you. If there is any doubt or query, feel free to contact me. Thank you. God bless you all.